So thank you. And first, good afternoon to everybody. I'm Aline Moreira Dias Martinez, and I'm from Brazil. And I just finished my PhD, as Meta said, at the Universidade de Brasília. And all my PhD was in association with Embrapa, Recursos Genéticos e Biotechnologia. And all my experiments I made at the Semiochemicals Laboratory. And our team is composed by Raul Laumann, which is my advisor, and Dr. Maria Carolina, and Dr. Miguel Borges. So this presentation, it's part of my PhD thesis, and the title is Multimodal Communication in the Neotropical Brown Stink Bug, Euschistus, Interaction between chemical and vibratory signals. Okay, first, just here in Brazil, the brown stink bug, it's a very important pest in soybean field. So because of this, we've been studying this insect for many years. And part of this study, it's about these insect interactions, but not with other organisms, but their reproductive behavior. And they already have identified and categorized the behavior. So, and part of their sexual behavior is about the sexual communication. And this sexual communication are mediated by some signals, as the chemical signals that have been identified by Miguel Borges and Aldrich, and the male sexual sex pheromone, the cuticular hydrocarbons, and the physics signals like the vibrational signals that it was identified before. And all the sexual behavior had been how I said before, characterized, and we have some steps. And first, we have the long distance communication that's mediated by the pheromones, and then we have the short distance communication that is mediated by vibrational communication. And then still in short distance, we have the contact, semiochemicals and visual signals, and we have vibratory signals too in this, the last step of their reproductive behavior, finalizing with the mate. And we know all this behavior, but the objective, the goal of this work is to understand, to see how the vibratory communication can interfere on the pheromone communication, and how these pheromones can interfere on vibrational communication, and how contact chemicals, that is cuticular hydrocarbons, can interfere on the vibrational communication. And to test this, we have three hypotheses, and the first one is the contact with chemical traces, and a chemical trace is the cuticular hydrocarbons that the insect left in the substrate by their tarsus. So if these chemical traces of the stinky bug influence on vibrational communication of the stinky bug, and this influence depends on the density of the trail. And the second is the vibratory signal from individuals of the same species interfere with the emission and composition of the sex pheromone. And then if the contact with male sex pheromone interferes with the vibrational communication, stimulating the emission of vibratory signals, and if influence the temporal and spectral parameters of the signs. To do all the experiments, we keep the insects at the Embrapa, Recursos Genéticos and Biotechnology, and all the experiments are made there too. So in acclimatized rooms, some experiments are performed in bean plants. So we cultivate these bean plants in greenhouses, and we use adult to 10 to 20 adulthood age or feeded and virgins. And as I said before, we use the brown stick bug Euschistus. Here's just to show the equipment that we use to do the experiments. As we say, to reproduce, we use a mind shaker, and to capture all the signal, we use a laser vibrometer. Well, the parameters that we analyzed is the latency, the response rate, the pulse duration, the interval between pulses, the dominant frequency, number of pulses, and mate proportion. And to test our first hypothesis that if the chemical trace can influence the vibrational communication, we made this experiment. To collect the tracks, we put 5 insects inside a Petri dish, and we put the Petri dish in this position to avoid the insect to escape, to keep them in the Petri dish, and to avoid any contamination. And they left their traces and this Petri dish. So we take off the insects and we put the Petri dish together, and then we put insect inside this arena, and then we put here, I don't know if you can see, but the reflective tape to record, and all the behavior was monitoring by real time. And to record, we use a laser vibrometer, and all the behavior was recording during 15 minutes. We made with single males, single females, and with couples. And I'll show just the most important results. And here we can see is this response rate. So when we can see that just the female made some difference in between treatments. The blue one is the control, and the orange is female, is arena with female traces, and this arena with the gray one, it's arena with male traces. So we can see if the females are in arena with control, that it's no trace, it's just the clean Petri dish, or in the Petri dish with the female traces, none of the females respond, and we say the response is emit a vibrational signal, but 25% of the females emit vibrational signals when in the presence of the male traces. These blood spots, we can see there's difference when the couples are in the female traces. Here is the number of mates. So in blue is when couples finish the bioassay with mate, and the orange one is when they finish the bioassay with no mate. So when the couples are in the arena with female traces, more of insects finish the bioassays with a mate. So we can say that with the presence of female traces stimulate the couples to mate, and the male traces can stimulate females to sing. And so we want to see if the same things can happen when we have natural substrate. For this, we keep 10 insects of the same sex in a part of a bean plant, and to keep them, we cover this part of the plant with a plastic bag, and we left the insect there for 2 hours. Then we take off the insect and we put here just one individual, And then we note all the behavior. We record here, as we can see with the vibrometer, all the vibratory behavior. And we noted too the initial choice. That means which side of plant the insect goes first, and the residence time, that how many time they spend in each side of plant. The treatment, that's plant control. That means plant with no trace, just the plant, and plant impregnate with male traces, and plant impregnate with female trace. And we can see here that the two first box plot here, we can see that the males, when they are in presence of plants treated with the female trace, the males goes to firstly to the stem that was impregnated with the female trace. In blue, it's the stem with treatment. The orange one, it's the stem with no treatment. And here is the control plant. So none of the side of plants are impregnated with the female trace. But as we can see, now the male, even the female, didn't show any preference for each side. But here, males prefer to go into the side that there was female traces. And when we see here the residence time, just the males, we can see that in control plants there are no difference between sides, but here in orange we can see is the side with treatment. So insects prefer to spend more time for running on the stems, the sides that the female's traces volume. So first interesting behavior is that the female's traces attract and attract and keep the males in places that their sexual partner traces are in the substrate. Here we will start to talk about the vibrations results with the vibratory behavior. And here, as we can see, the yellow ones, it's treated plants, and here the orange one, it's plant with control, that's no treatment. So we can see the females respond that emitted more vibratory signals in presence of plants that have the presence of the male's trace, as we can see here. But for male, there is no difference between the two plants. Even in control or in treatment, the names start the same. Here, just 21% of males emitted the vibratory signals, and 67% of the females emitted vibratory signals, and the plants with treatment. And here, when we analyze the males of Aerus responses to female traces in plant, here's just the behavior of males. We can see here in the first graphic here, and that the latence time was lower when the insect was in a plant with the female traces. The number of pulses was higher in plants with treatment too, and this pulse duration was lower, and the interval between pulses was lower too, as we can see in this graphic and this one, and the presence of the female traces. So we can see that the males are stimulated to emitted signals, and in the presence of this females traces. And one interesting behavior is that when the males was in the plants with no treatment, control plants, none of the males emitted the signal M3. And this signal was described in literature where that is a signal when the male gets in contact with the female. Just when the male get in contact with the female, or when they are doing the attenuation behavior, is where when the male rubs the antenna in female abdomen, they emit this signal. But here, when the presence of just the cuticular hydrocarbons of the female is present in plants, that stimulate the male to emit this signal. So we can see that this is very important to them, and the chemical, the hydrocarbons, cuticular hydrocarbons of these insects can interfere on the vibratory communication. Well, to see why this happens, we just identified their profile. The female profile has been described before, but we want to see if different from the male. So we put 50 individuals of the same sex in a pot with a Petri dish, covered with a Petri dish for 24 hours. Then we washed this Petri dish with hexane, and during this process, we rub it with a glass pipette during 30 seconds to get more hydrocarbons present in this Petri dish. Then we get this extract, and we just prepare the sample to inject in a GCMS with chromatograph, gas chromatograph, to the identification. And we find out that we have a primary identification, but here we can find out that it's different. Females have more compounds compared to males, and we can see that their chemical profile is different because some compounds are different present in female either cuticular, it's different from male cuticular. Okay, to test the second hypothesis, if the vibratory signs from individuals of the same species interfere the emission of sex pheromone, we made this experiment, and here we just connect the minus checker to a bean pod, and we recorded the calling song of the females from Euschistus called FS1. So in NS control, there's no playback to recording, and we get this arena, and here to keep a flow inside, airflow inside, we connected to a pump vacuum, and in the entrance of the air, we put activated charcoal to clean the air, and then we connected to SPME fiber to collect all the volatiles, and we collect during 30 minutes, and then we put this fiber in a GCMS to the quantification, and we quantify just the sex pheromone. And here, as you can see, this graphic shows the pheromone, the quantity of pheromone for insect during 30 minutes. So as you can see, when the males are stimulated with the female chromosome FS1, like described here, the quantity of pheromone was higher compared with the males, the collected of males in control arenas. Okay, and to test the last hypothesis, if the contact with male sex pheromone interferes with the vibrational communication, stimulated emission of vibratory signals, and influence the temporal and spectral parameters of these signals. We made this experiment. First, we get some struct of the male pheromone with a volatile collection system. We get this volatile and we per concentrate to equivalent of 1 insect for 5 microliters. Then we put this sample in a filter paper. Then we put this paper next to a bean plant, and during 15 minutes, all the behavior and the vibratory behavior too was noted and recorded during 15 minutes. We made with single males, single females, and with couples. And as control, we just put hexane in the paper to be the control of the experiment. Okay, this first one is the response ratio, and here the darker gray we can see is the plants with pheromone, and here in this bar with the lighter gray is the plants with just the hexane. So we can see that the response was higher of the single females was higher when the presence of the pheromone, and the others there's no difference. Here we can see the latence. So the females spend less time to start to emit the vibratory signals in plants with pheromone compared with plants with hexane. And in couples, the male spend more time to start the vibratory behavior when the pheromone was present in plant. Here, as you can see here in this last bars. The response time of the females when they are tested singly was higher too, as we can see here in plants with pheromone compared to plants with just hexane. Here's the number of pulse, and the males emitted less number of pulses during all their behavior, vibratory behavior in plants with pheromone compared with plants with just hexane. And here we have the buzz behavior. It's the, how can I describe as the wing wiggle, I don't know, but here we can see the females emitted more number of buzz in plants with pheromone compared with plants with no pheromone. And the experiment with couple, the male spend more number of buzz compared with the plant when in plants with pheromone compared with plant with no pheromone. And the pulse duration of the single female was higher when they're in plants with hexane compared in plants with pheromone, and males when they're tested as couple, the pulse duration was higher too in the female in plants with the hexane compared with plants with the pheromone extract. And the interval between pulses was higher in plants with pheromone, in males test singly, and with a male when they're tested as couple. Here is just is the results with couples, tests with couples, and here we can see that the proportion of vibratory the proportion of the insects that sing spontaneously was higher when the pheromone was present in case of the females. So when the pheromone was present, the females start to the vibratory behavior, and when the pheromone was not present, most of males start this behavior. And in a mate proportion, there's no difference here, as we can see in the second graphic. And we can conclude here is that the stinky bug chemical traces are recognized by their conspecific, and they can interfere with the vibrational communication and sexual behavior of the brown stinky bugs, Euschistus. The second conclusion is the emission of Euschistus female vibratory signals, stimulate males of their species to release their sex pheromone. And the last one is that the sexual pheromone presence can stimulate or stimulate in case of males, inhibit in case of males, the emission of vibrational signals of the stinky bug Euschistus eggs. I would like to say thank you to Embrapa and the Laboratory of Semiochemicals for letting me do all the experiments, and Universidade de Brasília to let me do my PhD, and all these agencies of our Brazilian government who give us financial support, and it's very important to us here. And thank you to everybody here that is listening